This is the review for Unit 3, Diffusion and Osmosis. So this unit looks at how transport happens across a membrane. So we always need to get stuff into a cell and stuff needs to leave a cell. Also, water needs to enter a cell or, in some cases, leave a cell. So it does this through the process of diffusion or osmosis. When we're talking about diffusion, we're talking about the movement of the solute from an area of high concentration to an area of low concentration. And the solute will always naturally want to move down its concentration gradient. So for example, the blue stuff here represents the solvent. If I pour some of the solute in here, the stuff that's being dissolved, if I pour it in there, you know that naturally it's going to want to distribute itself evenly throughout the solvent. So naturally it's going to move down its concentration gradient. The first objective asks, how does temperature and concentration gradient affect diffusion rate? So let's look at this. You actually are very familiar with this. If you were to try to make tea, for example, you know that it's easier to make tea whenever the water is hot. And the reason that is that when temperature increases, the molecules actually pick up speed. So the water molecules are bouncing off of each other a lot quicker. And therefore, it's easier for things to spread or diffuse. So a higher temperature increases the rate of diffusion. If the temperature is really low, diffusion takes a very long time for it to usually happen. Let's look at the effect of concentration gradient on diffusion rate. So one thing that you want to know is that as you increase the concentration gradient, and this means the difference between the concentrations, it's going to increase the rate of diffusion. This makes sense. If you have a greater difference in the concentration, then more stuff has to move in order to create an equilibrium to balance out the concentrations. So the greater the difference, the greater the rate. If you have a smaller difference in the concentrations, or in other words, smaller concentration gradient, then you slow down the rate of diffusion. One mistake that a lot of students make is that they say that they forget to add this word gradient. And that's what's important. We're talking about the difference between the two concentrations. So if you just say increasing the concentration increases the rate, well, that's incorrect. It's actually increasing the concentration gradient that increases the rate, so the difference. So let me show you an example here. What we have here is in each one of these, actually, we have one concentration of sodium hydroxide on one side of this membrane and another concentration on the other side. In A, which direction will sodium hydroxide want to move? It will want to go to the left. In B, it's going to want to also go to the left down its concentration gradient. In C, it's going to want to move to the right down its concentration gradient again, and also in D, it would move to the right. Notice that C has a higher concentration than D. However, it's not the concentration that really matters, it's the difference in concentration that affects rate. So D would have the highest rate because notice there's a 50% difference in the concentrations, whereas in C, this has the slowest rate. There's only a 5% difference in the concentrations. Objective 2 says, describe the role of the cell membrane in regulating the movement of materials into and out of the cell. Now this is really important because the cell membrane is selectively permeable. What this means is that only certain substances can move across the membrane and other things cannot. There are actually really small substances like carbon dioxide and oxygen and glycerol molecules. These are small nonpolars, so they have no charge to them. These molecules can actually squeeze right through the phospholipids, right through the cell membrane. However, you may have larger molecules that can't get through the membrane at all. Or you may have some molecules that have a charge to them, and they therefore have to use these purple proteins to actually be transported across the membrane. So this membrane can regulate what goes in and out. You did an experiment to test this in lab. And what you did here is you had what's called a dialysis bag or dialysis tube that had many tiny pores on it. In the inside, or on the outside, you had iodine mixed with water. And in the inside, you had glucose and starch. And the whole point of this experiment was to see if glucose, starch, and iodine could cross that membrane. And what you noticed at the end was something that looked like this. And we know that iodine and starch, when they come in contact, that it turns the solution black. So what this means is that iodine had to go into this bag and interact with the starch in there and turn the inside black. Did starch move out of the bag? Well, if starch would have moved out of the bag, then we would have expected the outside solution to also turn black. So we know that starch never moved out and only iodine went in. You also tested this beaker solution here for glucose. Remember, glucose started in the bag, and you looked at the results of this experiment with a little glucose test strip, and it turned positive, meaning that glucose went out of the dialysis bag. So why did we see this difference? Why was glucose and iodine able to move and starch was not? This all had to do with particle size. So glucose molecules are actually a single sugar molecule. They're pretty much one of the simplest forms of sugar you can think of. Okay? Whereas starch is composed of many glucose molecules attached together in this long chain. So starch molecules are really, really big. So starch could not move through this membrane because starch is too big. However, glucose could move through this membrane. So iodine also is very small, so iodine could easily go through the membrane as well. Objective 3 asks you to recognize how water potential affects the movement of water across membranes and note the factors that affect water potential. So when we're talking about the movement of water across a membrane, we are talking about osmosis. And water always moves. It's always going to do this, so just memorize this. Water always goes from a hypotonic solution to a hypertonic solution. And what this means is that water's always going from an area that has lower solute concentration to an area that has a higher solute concentration. So as you look here, also notice that a lower solute concentration has a higher water potential. And what this means, and one way to help you think about that, is that it's more likely or has a higher potential for water to move out of that solution or to leave it. Okay? So we know that water always goes from a lower solute to a higher solute. So the lower solute has a higher water potential, a higher potential for water to leave it. Whereas a higher solute concentration has a lower water potential. Water doesn't want to leave a hypertonic solution. If both solutions were equal in solute concentration, then we would call them isotonic. Let's look at an example. So let's say that I start out with something like this glass tube here. There is a selectively permeable membrane in the middle. There's a lot of solute over here represented by the purple and very little solute over here on the left side. So the left side would be described as a hypotonic solution because it has lower solute. The right side would be a hypertonic solution. Remember what I said before, water always, no matter what, will go hypo to hyper. So what does that mean? Well, it means that water is going to go into the area that is hypertonic, has more solutes in it. So let's see what happens here. What you see is that over time, the water will go to the right side and the water level will rise on the right side. And this is an example of osmosis. Objective number 4 says describe the tonicity of solutions. So whenever you're asked to describe the tonicity of a solution, do not say it has a lot of tone or it has a little tone. That's not what we're looking for. We're looking for you to describe it as either hypertonic, hypotonic, or isotonic. And you can tell whether it's hypertonic, hypotonic, or isotonic based on either the direction water is moving or on the solute concentration. So let's look at an example here. So here I'm looking at the movement of water, and I have 3 bags soaking in a 30% salt solution. Water can go through the membrane on the bags, however salt cannot. So I want to know which way will water move and which solutions are hypertonic, or hypotonic. So if we look at the first bag, there's 50% salt solution inside the bag. Remember, the water always moves from an area of lower solute concentration to an area with more solute. So in the first case, water will go into the bag. So is the inside of the bag hypotonic or hypertonic? Well, if we remember that water always goes from hypo to hypertonic, that will make solving these type of problems a lot easier. If we look here, we know that this side of the arrow is hypotonic and this side is hypertonic. So the inside of the bag is hypertonic relative to the outside. Also, we can note that a hypertonic solution always has a higher solute concentration than the other solution. Let's look at the second bag. Which way will water move in this case? Well, water will move from an area of low solute to an area of higher solute, so water will move out of the bag. So what is the tonicity of the inside of the bag compared to the outside? Well, the inside of the bag is hypotonic relative to the outside because water always goes from hypo to hyper. So what's interesting here, and I want to point out, is that the 30% salt solution is hypotonic relative to the 50% salt solution and is hypertonic relative to the 5% salt concentration. So whenever you're talking about hypotonic or hypertonic, you're looking at comparing 2 different solutions. In the last case, the solute concentration is equal, so water will move equally in and out. The inside of the bag would then be considered isotonic relative to the outside of the bag. So one reason that students get these questions wrong is that they forget whether we're talking about the movement of solute or the movement of water. And you have to first ask yourself that when you read one of these questions, because the solute is going to move in the opposite direction that water will move. So water moves from low solute to high solute. The solute itself, or the stuff in there, will always go from the higher concentration to the lower concentration. So in our first bag, which way will the salt move if it could move through the membrane? Well, it's going to go from the 50% to the 30%. Notice it's the opposite way that water moves. So which way in the second bag will salt move? Well, salt will move into the bag, down its concentration. In the last bag, this salt's going to go equally in and out, okay, because it's equal both ways. So just anytime you get to one of these questions, ask yourself, are they talking about the solute or are they talking about water? The next objective asked us to recognize the role of the cell wall and the large central vacuole in producing turgor pressure to maintain the shape of plant cells. The presence of the cell wall allows plants to take in more water and not burst. Okay, so they can actually maintain this large amount of water pressure. So if a plant were to be placed in a hypotonic solution, well, let's just think about that. So if it's hypo out here, what's going to happen to the water? Well, the water is going to go in. Water always leaves a hypotonic solution. So water will go into the cell. And what we would see here is that water would enter the vacuole, the large central vacuole here. And that vacuole is responsible for storing all that water, and it will push out on the cell wall and therefore increase the water pressure in there and make the cell what we call turgid. If that cell was placed in an isotonic solution, then water would go equally in and out of the cell. Notice how the cell wall isn't really pushing out against, the cell membrane isn't really pushing out against the cell wall because that vacuole isn't completely filled. If placed in a hypertonic solution, what's going to happen? Well, again, if you're ever solving a problem like this, just draw a square to be the cell. You know the outside's hypertonic. Well, water always goes hypo to hyper, so water would leave. So what does that mean? Well, the central vacuole would get really, really small. So the water has left, and we call that flaccid. This is why plants wilt. They start to wilt when the vacuole is empty and there's no pressure against that cell wall. Well, animal cells, they don't have a cell wall. So what typically happens when they're placed in a hypotonic solution is they'll actually burst because they have no cell wall to hold in all that water. In an isotonic solution, that's normal for a red blood cell or cells in your body. They want to be placed in a solution that has an equal solute concentration to the inside of the cells. And if an animal cell is exposed to a hypertonic solution, it's going to become all shriveled up because the water will leave. So here's a sample problem based on this. Which of the following plant cells were placed in salt water? Well, we know salt water has a lot of stuff in it, so the water is going to leave the cell and go into the area that has a lot of stuff. Well, this one has a full vacuole. This one's pretty full, so it means water's entering it. This is the only one that has a vacuole that's really, really small. So what that means is that the water left and went into the salt solution. So the answer here would be this one. Let's look at a question where it's asking about water being placed in distilled water. Well, distilled water doesn't have any solutes in it. So any other solution would have more solutes in it, like the inside of the cell. So water would enter the cell, because out here would be distilled. So you're looking for the picture that has the largest vacuole, because there's solutes in here. This vacuole isn't just filled with water, it's filled with a lot of different stuff. Okay, so water would move into it. Let's look at this last example here. This is one in animals, specifically people, and this is a real life situation here. People with a condition called hyponatremia have lower than normal sodium concentrations in their body, causing the inside of their cells to have a higher solute concentration than the fluid surrounding their cells. What would be the tonicity of the inside of their cells? Or what would the tonicity of the inside of their cells be compared to the outside of their cells? Well, let's draw a little picture, because I always love to draw little pictures. So here is the cell. Well, the inside has a higher, it says the inside of their cell has a higher solute concentration than surrounding it. So if it's higher solute in here, so higher solute, I know water will go into that. So this inside part is hypertonic because water always goes from hypo to hyper. Okay? Or you could have just remembered that a higher solute concentration means that it's hypertonic. So there's a lot of different ways you could have thought, a lot of different thought processes you could have taken to answer that question. Let's look at another example here. Same thing, but now it's asking us which of the following statements is true about someone suffering from hyponatremia. So it's asking about whether water would go in, whether they would lose water, or so forth. Well, as we just saw in that last example, if there's more solute in the cell, water would go into it. So let's look at the first one. There would be an equal amount of water flowing in and out. Well, that's not right. Their cells would lose water and shrivel up. Nope, we know water's going to go in there. Their cells would fill with water and potentially burst. That sounds pretty good there. But let's just read this last one just in case. Their cells would fill with water and experience turgor pressure. Well, it's definitely not turgor pressure because that only occurs in plants. So C is correct. Because they don't have a cell wall, water will enter and they'll potentially burst. All right, that is the end of Unit 3.